For those people that maybe don't think so much about treponemes as I do, there are 4 pathogenic treponemes of human, of which the most important is obviously Treponema pallidum subspecies pallidum, which is the cause of venereal syphilis, probably still the cause of several 100,000 stillbirths a year, and a very neglected disease really. Then the one I mostly work on, Treponema pallidum subspecies pateniae, which is the cause of yaws. This is the second most common of the human treponemal infections. And then these 2 at the bottom, Treponema pallidum subspecies endemicum, the cause of bedule or endemic syphilis. And then the last one, which is the only one that's not yet called a subspecies of T. pallidum, which is T. carachuum, which causes pinta, which is found in Latin and South America. This almost certainly really is a subspecies of T. pallidum, but there's no genome sequence available to allow anyone to rename the organism. And these bottom 3 are endemic and non-venereal treponemes, which are on the kind of approved WHO list of 17 neglected tropical diseases. And so yaws, which is what I'm going to focus on, it's found mostly in warm, humid environments around the world, and the kind of major foci these days are in West Africa, South America, the Pacific, and Southeast Asia. Unlike with venereal syphilis, children are the main reservoir of disease in yaws. So children aged 5 to 14 are where most incident and prevalent cases are found. Transmission, just like in syphilis, is skin-to-skin contact. It's just not skin-to-skin contact involving your genitals. It's skin-to-skin contact normally involving your arms or your legs. And I think, an interesting thing is this is the first disease ever, before smallpox, before guinea worm, before anything else, to receive a World Health Assembly resolution for eradication. So the first call to eradicate yaws was in 1948, and it's 2016, and I'm giving a talk called Towards the Global Eradication of Yaws, which might lead some of you to question how successful those previous calls have been. So what does the disease look like? So like syphilis, it's a multi-system disease and a multi-stage disease, and after inoculation, people develop either these papillomatous lesions or these typical-ish ulcerative lesions, and classically these are on the lower limbs, but occasionally on the face, the arms, can occur anywhere at the site of inoculation, but normally the lower limbs, more than 80%. If it's untreated, you get hematogenous dissemination of the spirochetes and lymphatic spread, and as in syphilis, you can get basically a very broad array of rashes. So you can get rashes on the palms and the soles, you can get multiple ulcers over your body, you can get multiple papillomas, and you can also get involvement of the bones and the joints. So dactylitis is very commonly seen in Papua New Guinea, which is one of the real hotspots. If you're treated at this point, these lesions go away, you're cured, everything resolves, your skin re-epithelializes, the bones go back to normal. It's a very, very good outcome. If you're not treated, you can progress, just like in syphilis, after a number of years to these very destructive tissue lesions of tertiary yaws. There's nothing you can do about this. You can cure the person of their spirochetal infection, but obviously the disability, the disfigurement has occurred. And so by intervening early, this is really what we're trying to get rid of. I mean, a skin ulcer is not a very pleasant thing, but it's also not that dreadful, but obviously we'd like to avoid this as an outcome. And just to show you what happens with treatment, so this is in a trial in Ghana that we're running. This is at week 0. 1 dose of treatment, the lesion's re-epithelialized. 6 months later, you can barely tell that someone's been treated. So you can get a really good result if you treat people early. There's nothing you can do if you miss this window. Like syphilis, you can have latent infection, so people who are asymptomatic but have spirochetes hiding somewhere in their blood, and this can only be diagnosed currently with serology, and these are really key to the eradication efforts because they are the most important source of reinfection. And there's a high ratio of latent to clinical cases, so maybe as many as 10 latent cases for every clinical case. So you can imagine a campaign that focuses only on active cases will be a very unsuccessful eradication campaign, and we'll come back to that. Why is anyone talking about yaws? I mean, to be honest, this is a niche disease. I say that even as one of the people that works on it. Probably really the only reason anyone's talking is this study, very nice study done in Papua New Guinea by Oral Mitya and colleagues. They randomized children with primary and secondary yaws to single dose of benzathine penicillin, which is the standard of care since the 1950s, or a single dose of oral azithromycin, and showed that oral azithromycin had a cure rate of 95% at 6 months. Which you go from a drug that you need trained staff, needles, and there's a risk of anaphylaxis, to a very well-tolerated drug which is already used in other NTD control programs, which can be orally administered by untrained staff. So there's some kind of sea change there in what's possible treatment-wise. And the WHO, in their kind of infinite wisdom, took only 6 months from the publication of this paper, start of 2012, to by the end of 2012, they had already formulated a new global eradication strategy for the disease, and there would be no disease in the entire world by 2020. So that will be 8 years from a single randomized control trial to worldwide eradication of a disease. Some people might say that that is slightly optimistic. So what does that strategy look like? It's called the Morges Strategy because of the place where it was come up with. And the idea was that you would do an initial round of community mass treatment using azithromycin as the new standard of care. And then you would go back every 3 to 6 months. This is a number that's effectively plucked from thin air, but seems like a plausible-ish number. And you would find the cases and their contacts. Think about, is that finding active cases? Is that finding latent cases? How are we defining a contact? We don't really know. And once you have 0 cases, active cases, we think, you'll switch to surveillance and you'll do serological surveys on children who are 1 to 5, because if you follow those people and there's no transmission, they should never seroconvert. You've interrupted transmission. Hooray. So just a little bit about why they went for mass treatment. As I mentioned, there have been many previous control programs, and probably the problem has been that one of the problems has been that they gradiated what you should do. So they said if there were more than 10% cases in the children, treat everyone in the community. If there's 5 to 10, treat the children and treat any contacts. If it's under 5, just treat the cases and the contacts. But obviously, they missed loads and loads of latent cases when they did this because they only identified predominantly active cases. There are 10 latent cases for every active case. You go away, the latent cases relapse to active disease, the disease comes back. So they almost certainly missed many latent cases and had inadequate post-treatment surveillance. And so unsurprisingly, although this was an incredibly successful campaign, if it wasn't considered an eradication campaign, they treated 300 million people, and the prevalence of yaws worldwide fell 99%. So if they had called this the Global Yaws Control Campaign, this would have been a success. But this was the Global Yaws Eradication Campaign, so one case is a failure, and they had far more than one case at the end. So just to show you that it did work in the places they tried, this is in Nigeria, 3 different districts, and the prevalence of yaws at baseline, mass treatment, 6-month follow-up, 12-month follow-up. I mean, it does work, it seems, as a strategy if you can get high coverage and identify the right people. And more recently, India is probably the kind of big success story. This is nothing to do with the WHO. This is a national program in India run by the government. And they have had a very aggressive strategy of finding cases and treating. You can see it took them from 1996 to 2004 to have 0 cases. That's obviously not as much time as we have to go from 2016 to 2020 to have no cases in the world. So that makes that target slightly tricky. But they were very successful. They did mass treatment. This is with benzophine penicillin. This is before azithromycin. They used a number of strategies, including paying people to report suspected cases of yaws in the community. And you got the rupees if the case was serologically confirmed, and you didn't get the rupees if the case wasn't serologically confirmed. But it worked. It seems to have worked. And they're now out at 2016, and their serology, there are no seropositive children under the age of 5. They look like they really have interrupted transmission. So that's the kind of upswing of the talk. That's a bit about yaws and how we're going to eradicate it, because India has achieved it. And I'm going to spend the rest of the talk talking about why we probably might not eradicate it. And there are, as you can see, some problems across the board. We don't really know the epidemiology of the disease, which might be considered a small problem if you're going to try and eradicate something. We don't really have access to the correct diagnostics, and we'll see that that's becoming more and more of a problem. We presume that mass treatment with azithromycin will work because mass treatment with penicillin worked, but maybe those people who remember azithromycin for other organisms might have some worries about that. We'd quite like some money to eradicate yaws. And then obviously there are the kind of Donald Rumsfeld things, the known knowns and the unknown unknowns. And as you start down the journey towards eradication, then obviously you uncover all sorts of things that you didn't know were going to be problems at the beginning, and which might turn out to be problems as you get kind of closer to zero. So some gaps in epidemiology. This is the current map of yaws in the world. It is, in my opinion, mostly made up. The countries in red are currently endemic. That means that they ring up the WHO once a year and say, we saw some cases of yaws this year. So there are, I think, 13 on the map. And they're mostly in West and Central Africa and in Southeast Asia and the Pacific. And that's almost all clinical case reporting without diagnostic confirmation and without active case finding. So this is passive case finding. People come to your facility, you think they have yaws. You can already see that many of those countries which are in orange, which are places that used to tell the WHO that they saw yaws and don't currently, might actually be red. And how likely you are to eradicate something might, for example, depend on whether or not there really is yaws in Sudan or Ethiopia or Kenya or all of Africa mostly. And then the two success stories which are in green are Ecuador and India, and they both achieved local elimination, it appears, with government-led programs, sustained benzathine penicillin use before this most recent WHO call for eradication. And I've spent the last kind of 2.5 years living down in the Solomon Islands, which is a kind of archipelago just off the coast of Papua New Guinea. So just to put some numbers on that, these are the number of cases that these countries say they see. So you can see that, I mean, although Benin, the whole of Benin is red on that map, they don't see very many cases. And you might like to know where in the country those cases are before you decide to give 10 million people azithromycin. Because if you can get away with only giving 500,000 people, that would be much more effective. So the big players are Ghana, reports about 10 to 20,000 cases a year. Papua New Guinea and the Solomon Islands, where I work, these countries all report more than 10,000 cases a year. And the number at the bottom is the incidence per 1,000 people in the country. So you can see that the Solomons is, by an order of magnitude, the place where there is the most yaws in the world. 1 case for every 2,500 people. Lots of yaws, so they say. So that's just to summarise, there are these 72 previously endemic countries. Either we need to find that there is yaws there so that we can go and eradicate it, or we need to certify that there really isn't yaws there, not that they're just not reporting it. The 13 endemic countries, we'd like to know where the yaws actually is in the country, how much, so that we can target our interventions. And we don't really know how we're going to do that. There's no agreed evaluation unit, and there's no money to do any yaws mapping. So that's slightly challenging. And people are taking other approaches, trying to integrate with other programs that are going out to do mapping for LF, or mapping for trachoma, or mapping for podoconiosis. Try and piggyback, basically, off other people's money. So we don't have very good epidemiology. Do we have very good diagnostic tests? The diagnosis is the same as in syphilis. 2 tests, your TPPA stays positive for life, RPR, VDRL, the titers go up during infection, they fall following treatment. It's not a specific test, but it's the only one that reflects response to treatment. Traditionally, you've needed molecular tests if you wanted to be able to tell the difference between T. pallidum pallidum and T. pallidum pertenniae. And you might think, for example, that there's quite a lot of syphilis in Papua New Guinea and the Solomon Islands, and you'd be right. And so when you're counting cases and saying this is a case of yaws in a 16-year-old, you'd really like to know it really is T. pallidum pertenniae, not T. pallidum pallidum, because we're not trying to eradicate syphilis, we're only trying to eradicate yaws. And that's really important because 1 case is a failure. So you have to know exactly what it is that you're counting. So this is an RPR, which I think is just a kind of incredible thing that anyone is still doing in the world. It's an agglutination assay. You put some serum, you put some stuff, you swirl it around, you see if there are some black dots. If there are black dots, it's positive. It's a great test. Anyway, we're hoping to replace that with this. This is a rapid diagnostic test, which was obviously invented for syphilis. There's no one inventing rapid diagnostic tests for yaws, for obvious reasons. But the serology is identical. So this test that works for syphilis should work for yaws. And the good thing about this test compared to other tests is it contains both a treponemal and a non-treponemal component, whereas most of the syphilis RDTs only have a treponemal component, which is not helpful when you're trying to eradicate a disease because you've got a test that stays positive for life, so you need a test that goes negative. And it's been evaluated in a number of settings. We've evaluated it in the Solomon Islands, colleagues have evaluated it in Papua New Guinea, and the CDC do have a multiplex PCR now that can tell the difference without sequencing between T. pallidum pallidum and T. pallidum petenae. So just to say it does work in real life, this is a negative test, the control line's positive, the other two lines are negative. This is old infection, so just line 1 has come up. This person's had yaws or syphilis at some point in their life and been cured, and this person has current infection, so both the treponemal and the non-treponemal components are positive. I can do this test, which means it's an incredibly simple test. It takes about 15 minutes, and we're using them in the field in a number of countries now. So maybe we've addressed some of the questions about diagnostics. We've got a point of care test, we've got a PCR test that looks like it might work. Will MDA with azithromycin be effective? This is probably the thing where we are having, shall we say, the most hope that we have the answer to some of our questions. This is data from Oriel Mitchell's group again in Papua New Guinea. They took an island, 20,000 people. They did mass drug administration with azithromycin, high coverage, more than 80%. And then they did follow-up surveys every 6 months as recommended and treated cases and contacts. And you can see by age group the fall away from baseline, those very high prevalences, to 6 and 12 months. The prevalence kind of really dropping dramatically, which fits with that data from India or from Nigeria, showing that if you get high coverage, you can bring treatment right, you can bring prevalence right down. Obviously, it's not gone to 0, and they're now at month 36, I think, and they still have incident cases. So either they are not treating the right people, or obviously they haven't treated the whole country, so there's infection coming in and out of those communities. And so that's a real challenge. This is one guy who has spent a long, long time and is probably achieving much better results than can be achieved programmatically in many settings, because it's a dedicated research team. And he hasn't quite got on top of it yet, but he's getting there. And we saw the same in the Solomon Islands. So this is cases of yaws by quarter for the time preceding mass treatment, and then you can see that mass treatment was conducted and there was a kind of rapid and sustained drop away in the number of cases seen. And we see similar prevalence data changes to seen in Papua New Guinea. So it does look like probably mass treatment with azithromycin is effective, with the caveats that we don't know what coverage we need. There's high coverage in Papua New Guinea and there's ongoing transmission. And we don't know how many rounds we need to go. And so it's quite difficult to turn around to Pfizer and say, can I have 100 million tablets of azithromycin? Because they say, how many doses would you like to give out? And I say, well, I don't know how many countries the disease is in. And how long would you like to give azithromycin for? And I say, 8 to 10 years, something like that. So that's a challenge in convincing someone to give you a drug when you don't know how much of it you need or for how long. We also don't know what dose to give. That's a bit of a challenge, particularly in the Pacific, where both trachoma and yaws are endemic. Some of you may know that there is a donation program for trachoma. It's at 20 milligrams per kilogram to a maximum of 1 gram. And for yaws, the recommended dose at the moment is 30 milligrams per kilogram for a maximum of 2 grams. If you think that latent cases are particularly important and that those organisms are probably replicating more slowly, you might want to get a really high dose into your macrophages to really kill those organisms. So you might think that the higher dose is better. And so that's a problem for countries in the Pacific who've got money and drug to do a trachoma program, because they don't want to give potentially a subtherapeutic dose for their yaws control and induce problems potentially in the future. So we are going to address this. There's an RCT, which I'm one of the investigators on, running in Ghana and Papua New Guinea. And it's a very straightforward trial comparing 20 milligrams versus 30 milligrams of azithromycin, with the hope being, obviously, that 20 will prove non-inferior, and therefore that will make life particularly easy for the countries in the Pacific, and might also make it easier to convince Pfizer to extend an existing donation program at the same dose, rather than setting up a whole new one at a different dose. We wait to see. Probably some people remember that azithromycin was touted as the new drug for syphilis. I mean, we use azithromycin for lots of other STIs. It's a single agent. You can give it. It's much nicer than giving an injection. And it was very successful in the original trials. Unfortunately, there is lots and lots of azithromycin resistance in syphilis now throughout the world, pretty much wherever you look. And it's conferred by just one of 2 single point mutations in the ribosomal RNA genes. So this is not a complicated resistance to pick up. no one has seen a azithromycin-resistant Yaws strain yet, but these organisms are 99.97% genetically identical. So clearly resistance will be inducible in Yaws. And whether or not we see resistance emerge, that will scupper the program because you'll have to go back to injectable benzathine penicillin and all the problems logistically of delivering that brings. So this is a real worry ahead. This was one of the known unknowns. Gaps in funding. The WHO have come up with a number. I take this number with a pinch of salt. They say it's $360 million to get rid of Yaws. They make some quite sweeping assumptions in this model. They assume that all 72 formerly endemic countries are truly not endemic for Yaws, and that it won't cost very much to certify those places. And as these models often do, it assumes that you will eradicate Yaws by 2020, and then you will accrue cost benefit for a very, very long time that you run the model in the future. So we'll spend $300 million now, and by 2050, we'll have saved X DALIs. Obviously, if you run any model very, very far into the future and you accrue a cost saving up front, eventually something becomes cost effective. So it's really highly dependent on a better understanding of the epidemiology in all the countries and on someone donating the drug. So that's the known unknowns. What about the unknown unknowns, the things that we didn't think would be problems but are problems? So one of these two lesions is Yaws, and these people are both in a Yaws endemic community, and these people are both seropositive on the rapid diagnostic test. You cannot tell these lesions apart. And this is not to me surprising. Why should all skin ulcers in a Yaws endemic community be Yaws, even if they're seropositive? But it is what people have been doing. They've been counting seropositive or even unseropositive ulcers in endemic communities and saying that's a case of Yaws. Obviously, that's a problem if you're going to eradicate it, if they're not all. And actually, overwhelmingly, they're not. So Haemophilus ducreyi, which people will remember as the cause of chancroid, seems to have gone away really as a sexually transmitted disease in much of the world, but is very, very common, actually, it turns out, as a cause of non-genital skin lesions in children in these communities. To the extent that in the bottom study, which we did in Ghana with funding from the Royal Society, we found 100 children with Yaws-like lesions in a place where azithromycin had been given. They were all seronegative. They were all PCR negative for T. pallidum. So these were 100 cases that would have been reported by the government into those numbers that you see as cases of Yaws, and none of them were Yaws. So this is a real challenge. And this is, again, data from Papua New Guinea showing the breakdown of what you find if you PCR these lesions. So these are cases that have been identified by probably the world expert in Treponemal pallidum subspecies Pateniae. He said that's a Yaws lesion, and these people are all seropositive. And what you can see is that at baseline, only 20% of them had T. pallidum only DNA in it. Another 13% had T. pallidum and Haemophilus ducreyi DNA in it, and far more of them had only H. ducreyi, almost 50%. So he is wrong more than 50% of the time when he says that that's a Yaws lesion. And by this point, he's done serology, so he's also been wrong, just as I'm wrong when I do this, on another 50% where he said that's a Yaws lesion, and the first test, the RDT, was negative. So we don't really know what we're counting anymore, which is a bit of a problem when you're trying to eradicate something and prove that you can count the number down to 0. You might say it doesn't matter. This is an incredible study. I'm glad I didn't volunteer for it. They randomized people to taking a single dose of ciprofloxacin or azithromycin, and then they injected them in the arm every week with Haemophilus ducreyi and found out whether or not when they came back the next week, they had an ulcer on their arm. And what you can see is that if you got injected and you took ciprofloxacin, you were in the good group. You got an ulcer very, very quickly, so you didn't get injected another 10 times. You didn't want to be in the azithromycin group because the median time to, the median number of injections was 6. So actually, azithromycin is a really good drug for Haemophilus ducreyi. It's a good drug for genital Haemophilus ducreyi. So if you do MDA in these communities, you do see that the Haemophilus ducreyi lesions go down, as do the T. pallidum lesions. They don't go away, but they do go down. But it's still tricky when you're trying to count something. Last of the kind of unknown unknowns, which I'll start on, it's a prerequisite, isn't it, of an eradication program that there isn't an animal reservoir. Because if there's an animal reservoir, then you almost certainly won't be able to get rid of this thing. So that's potentially a problem. So this is, I think, quite a nice map. This is countries in green currently report cases of yaws. Countries in grey have reported cases in yaws. We don't know if there's currently yaws in them. And then the red circles or stars are places where non-human primates, including both monkeys and small and larger apes, have been found to have either serologically or PCR-confirmed T. pallidum infection, causing ulcerative disease. So obviously, this is just kind of ecological data, but might lead one to worry slightly that there could be a non-human reservoir. And what happens if you sequence them is the top group, that's syphilis in blue, in green that's Bedgel, and then in red are the Yaws strains. And I would say to you that the two black ones at the bottom, Baboon Strain National Park, are as closely related to the human Yaws strains as the human Yaws strains are to each other. So genetically, this organism in baboons is indistinguishable from human yaws. So that's a slight challenge. And we know that if you take a bit of baboon yaws and you inject it into another lucky volunteer, they will develop a clinical lesion. And if you get a bit of human yaws and you inject it into a lucky baboon, they'll develop a lesion. So we certainly know that these strains are, at least in the laboratory, capable of infecting bidirectionally. We know that there's epidemiological overlap. We know that genetically they look like the same organism. So we don't really know if there are animal reservoirs elsewhere. Obviously, there are many, many monkeys in Southeast Asia and South Asia, and there's quite a lot of yaws there. So one might like to look at those, and we're doing that. And obviously, just because you can inoculate something in the lab doesn't mean that this is a real thing that's happening at the human-animal interface. But I think experience shows that, zoonotic transmission is very, very common, and the interplay here will be very complicated, and it might not be till we get down to very small numbers of cases that you start to detect more rare zoonotic events, as we've seen in other diseases like guinea worm. So I'm just going to summarise where we've got to. Some problems facing yaws eradication. We don't have any epidemiological data. We don't really know about the diagnostics. We don't really know if mass treatment will work. We don't have any money to pay for anything. And all the things that we didn't know were problems, but are pretty clear problems, like we don't know what a case looks like anymore, and we don't know if humans are the only reservoir anymore. So I think we've made some progress towards these two. I think, we've got a rapid diagnostic test and we've got a PCR test. So, rolling those out at a programmatic level is a different issue, but kind of intellectually we know what one could do, and it looks like mass treatment will work. We haven't addressed any of the others, which is a challenge. So we've made some progress, treatment and diagnostics, as I said. We've got big gaps, epidemiology, no money, non-human primate reservoirs, things that look like yours that aren't yours. So I'll conclude really just by saying that I think the WHO target of global eradication by 2020 is hugely, hugely ambitious. And I will say I would take anyone's bet that it won't be met, sadly. Thank you very much.